Hello, everyone. Sí, funciona. Thank you, everyone, for coming. We'll start. Maybe will come someone else, but we start. Today we have with us Cecilia. She holds a bachelor's degree in biological sciences and a master's degree in ecology by the University of Rio de Janeiro. She did her PhD at the University of Sao Paulo, conducting research at the interface between ecology and health. Then she moved to Portugal for her first postdoctoral position at the University of Coimbra. And then she got a Marie Curie to continue her research at the Complutense here in Madrid. And now she has an Atracción de Talento, where she keeps working on the interplay between ecological and evolutionary dynamics of species interactions and the dynamics of infectious diseases. And to do so, she combines empirical data, mathematical modeling, and tools from network sciences. And she will tell us everything about it. The floor is yours.
Okay, thank you, Nuria, and thank you all for the opportunity of coming here today and presenting a little bit of the work I've been developing these last 5 years together with the group that I work with, the students and postdocs. And while it's not a big surprise for everybody here in this room that we are living in an era in which we observe increased and accelerating trends in many socioeconomic and also Earth system parameters. So I would like to give special attention here that since the last, thousands of years, we are observing a great increase in the number of vertebrate species that have been already extinct or local populations that have been extinct. At the same time, when we observe the distribution of the mammal biomass in the world, here we also have this timeline and how these biomass distribution have been changing since these many years. And there is this great decline in the biomass of the wild mammals. Nowadays they represent only 2% of the global mammal biomass. But at the same time, there's a great increase in the biomass of livestock, which nowadays represents 65% of the total mammal biomass. And humans, they represent 35% of the global biomass. And at the same time, we're also facing a great increase in the area of domesticated land, which are agricultural land and also land used for urbanization and other kind of human anthropic uses. We've also facing a great increase in the abundance of invasive species, so species that start occurring in new habitats where they didn't occur before, and they have these characteristics of being invasive. And also a great increase in the wealth and size of the fortune of the great billionaires. And it is not a coincidence that the same pattern we observe in the cumulative number of zoonotic emerging diseases events. So zoonoses are those infectious diseases that are transmitted from wild or not, or from animals. They can be mammals, they can be birds, to humans. And those emerging disease events, they were mostly related to changes in land use and the intensification of food and agricultural industry. Here in this table, I'm just showing some examples of those emerging diseases. And just to be clear that when we talk about disease emergence, we're not only referring to new diseases that appear in the population, such as the coronavirus, this is a disease emergence, but also if we have a known disease, such as malaria, dengue, and they start occurring in different areas where they did it different when we consider the biology of those parasites because they can have or not different types of wild reservoirs of host species. They can have or not intermediate host and vector species. And they can have different mechanisms of transmission among humans. But what is a common to all those emerging diseases is that the primary drivers, land use changes is a primary driver that have been related to all of those events. There are other drivers, such as climate changes and agricultural intensification, but land use changes were related to all those events. And although we know what are the main drivers, we still have a very low predictive capacity on these events because we don't know exactly what are the mechanisms that link land use changes and the emergence of zoonotic diseases. And in this sense, when, as I said previously, if we're talking about disease emergence, we are talking about the expansion in the distribution of parasites in the sense of geographic distribution or in the sense of expanding their host range. So in parasite interactions, there are two main concepts that are related to their host range. So we have capacity space and opportunity space. This is going to define the fundamental host range, and locally we have a realized host range, so the host species that they are interacting at local communities. And in the case of the capacity space, so pathogen genetic diversity and plasticity, host compatibility, transmission efficiency, and virulence evolution, all those factors are related to the capacity space. And to the opportunity space is basically the probability that they're going to encounter. So host ecology and behavior, habitat structure, and environmental changes. Sorry. Okay. And what are the impacts of land use changes in these different spaces? So land use changes may increase the genetic diversity, susceptibility of potential hosts, and this is going to change the capacity of space of the parasites. And in the case of the opportunity space, we can change contact rates and the abundance of potential hosts. And all these effects of land use changes in those two capacity and opportunity is going to end up changing the realized host range of different parasites, which can result in spillover to different host species or expand their distribution range. So having this in mind, during the last 5 years, I've been working with this problem, and what I'm going to present here today are different questions we try to answer related to how land use changes can affect the diversity of host species, how can it affect parasite evolution, what would be the expected effects in the dynamic of host-parasite interactions, and also how the socio-ecological vulnerabilities can affect the risk of disease emergence. And for answering these questions, we focused in the Brazilian territory, and this is because it is a global hotspot of host diversity. Here's the distribution of mammal species richness. We can see that Brazil is one of these hotspots. It is also a hotspot of different species that have zoonotic parasites. So here we have the highest number of species that have been observed containing or hosting zoonotic parasites. And it's also a hotspot of land use changes. So here are the global major drivers of land use changes, and we can also see that in Brazilian territory we have a great diversity of most of those different drivers of land use change. And for those who are not particularly know what are the different patterns that those drivers leave in the landscape. So commodity-driven deforestation, we would have these great large spots of deforestation. Shifting agriculture would be more fragmented together with more vegetation cover. This would be the pattern left by forestry, wildlife, and urbanization. And for asking the first question, so how it affects the diversity of host species, we focused in the Atlantic forest, that is this biome that occur here in the coast of Brazil. And in each one of these dots, we have the entire structure of the small mammals communities that occur there. And we also have then the landscape use and configuration. And we use this data and joint species distribution modeling, in which we also incorporate not just the community composition, but also species traits, landscape structure, and the phylogeny of the host species to better understand how land use changes are affecting those communities. And these are some results. So here we have the meta-network, so the Atlantic forest network of different zoonotic parasites and mammal host associations. The size, so the squares are the parasites and the circles are the hosts, and the size of those nodes are related to the betweenness centrality of those species in the network, so related to the, depending on the betweenness centrality, we were able to separate, characterize those host species if they are central, if they have high betweenness centrality, if they're not central, or if we don't have registry, meaning that we don't have information about their interactions with parasites. And when we use this modeling framework related to different land use drivers, we found that the central species, they have a much higher probability of occurrence in places where you have a lower vegetation cover. They also have high probability of occurring in places where you have a higher percentage of farming landscape that is, can be both agriculture but also livestock creation, so cattle. And they are also highly probability of occurring in places where you have highly fragmented landscapes. And when we investigated how these co-occurrence of the host species are related to how they share parasites in the network of associations, this network is exactly the same network I showed previously. The only difference is that now I organized it in the way that I want to highlight the different model of species that this network have. So we have 6 different models. And we correlated the parasite sharing of those mammal species in this network of parasite-host interactions with their co-occurrence in the landscape. And this is the matrix of correlation. And what we found is that there are some models of host species that they have a high probability to co-occur but they do not yet share many parasites. So they co-occur, they are co-occurring in the landscape, but they not yet are sharing parasites because they don't share parasites in the host parasite association network. So what this is indicating us is that in those models, there's a high opportunity for parasite sharing there, but they not yet are sharing. So those are the potential species that probably depending on the circumstances, they could start to share parasites. And when we investigated for all mammal species and zoonotic parasites that occur in Brazil, we applied a similar approach considering the traits and host traits and phylogeny to calculate what would be the probability that they're going to share parasites, not yet, but that they're going to share parasites. Here is the probabilities. If it's gray, it's because they already, that parasite already interact with that host species. And we found highest probabilities in the primates and Chiroptera others. So those groups of mammals would be the ones that would have a high probability of starting to host those different zoonotic pathogens. That they are virus, they can be, and also different bacteria and protozoa. So what these first results are indicating us is that fragmentation and agricultural intensification favors key reservoir of zoonoses, and that high co-occurrence probability among host species that do not currently share parasite fauna may indicate potential hotspots for future parasite sharing events. So we now ask ourselves, okay, but then how land use changes affect parasite evolution? And when we talk about evolution, there are two main processes that lead to evolution. So we would have migration and genetic drifts, which would lead to geographic isolation among populations, and then we can start finding differentiation among those different species. And we also have natural selection, which would lead to local adaptation and also increase or not differentiation among populations. And in the case of this question, as a first general approach, I focused in this neutral process, that is the process of migration, to try to understand how parasite evolution would be affected by fragmentation. And then for doing that, I applied the Rida-Higgs model for metapopulations, that is a stochastic model, population differentiation. So each parasite individual is characterized by a genome, and it's a biallelic. That means it had two losses, so it's a string of ones and zeros, and this is characterizing the individual parasite genome. And the similarity between two individuals is what determines if they are belonging to the same species or not, because then you have a threshold, and above this threshold, those two individuals, they're not able anymore to mate, and they're already considered a different species. And it's a metapopulation, so it's spatially explicit, and in each patch, we have a local population, and the connections among the local population is determined by what is going to generate the gene flow among this population, and it's related to a migration probability. So we applied this model in different landscape scenarios, and how did we construct the parasite metapopulation? So we have this landscape in which, well, we have the green patches, they are not habitat for the host species. The purple ones would be potential habitat for the host species, and we have currently populations in these red squares. And we connect the host populations if their home ranges overlap. And if the home ranges overlap, then you have potential flow of individuals there and potential transmission of the parasites. And then this is how we construct the parasite metapopulation. Those dots here, the nodes, they are local parasite populations, and they are connected because then you have a flow of host individuals that is connecting those populations. And then you have also a gene flow there because you have flow in individuals. So this is an example of a parasite metapopulation considering a landscape that is pretty much continuous, so it's a very connected metapopulation. And if we go to a very fragmented landscape, then we start also fragmenting these parasite metapopulation, and we start finding local populations that become isolated or models of populations that are isolated. So here are some results. We explored different scenarios of habitat fragmentation, going from low fragmentation to high fragmentation, but keeping forest cover the same among all these scenarios. So we are investigating here how fragmenting this metapopulation leads to the different evolutionary trajectories. And when we look at the resulting parasite diversity, this is fragmentation by the number of parasite lineages we find there. We see that more fragmentation increase the number of parasite lineages. And this variance here is the variance in the different sizes of host home ranges we have there, which is an indicative of like a diversity of different hosts. So if you have only one home range, you will have only one host species. But if you increase the variance of this home range, you will have different host species in this landscape. And the interesting also to see that this decreases. So the highest the number of different host species, we will have also reducing the diversity of different parasite lineages. And the pattern is pretty similar when we look at the heterogeneity of this diversity across the landscape. So a measure of genetic FST has exactly the same pattern, so fragmentation would increase it. So we found that so fragmentation increased parasite diversity, enhancing their adaptive potential and potential capacity to colonize new host species, and also that higher diversity of hosts limits this diversification. Finally, we wanted to explore what would be the effects on the dynamics of those host-parasite interactions. And in this case, we used a spatially explicit eco-evolutionary model in which we considered not only evolutionary processes, but also ecological dynamics. And in this case, we also included the process of natural selection and transselection pressures. So this is the general framework used in this eco-evolutionary model, in which we have the ecological part of the dynamics is a meta-community dynamics. We have a landscape. In each patch of this landscape, we have a local network of hosts and parasites. And the meta-community dynamics is coming from a regional network of potential interactions and host and parasite diversity. And locally, we have the realized local networks in each one of the patches. And the meta-community dynamics is changing the different occurrence patterns of those species and interactions. And the second part of the model, that is the evolutionary dynamics, is considering coevolution between the hosts and the parasites. And there is a feedback between the ecological and the evolutionary processes. In the part of the ecological dynamics, it's the dynamics of extinctions and colonizations. In the case of the host species, we consider that they have the extinction and the colonization probability is related to the landscape structure. So if it's a species that is pretty much linked to forest cover, so would have a higher probability of colonization patches that have more forest and a lower probability of getting extinct. And this changes according to parameters that we put for individual species. So how are they affected by landscape structure? And in the case of the parasite species, their extinction and colonization probability depends not on the landscape, but actually in the matching they have with their hosts. So it's a trait matching pattern. And also, so the extinction decreases. So there is a high extinction probability if they have a low matching with their hosts, and they get extinct if there's no available host in the patch. And they have a higher probability of colonizing patches where they have a higher number of available hosts and where they highly match those hosts. And they can only colonize a patch where an available host is present. And in the case of the evolutionary dynamics, so we have a local coevolutionary dynamics. In each one of those local networks, we have an evolutionary dynamics. And this is, well, a model that have been developed previously. And so each species is characterized by a trait. As I said previously, a trait is a value that characterizes its trait. So it's Z. And it's, they are responding to two different selection forces. So environmental selection that is related to the matching of their traits with the trait that is selected by the landscape. So if they are favored by forests or if they are favored by disturbed places. And in the case of the species interaction selection, it's according to the interaction with the parasites. And for the parasite, it's favoring matching with the trait. So they want to have a trait that is matched to the trait of their hosts. And in the case host species, they want to run away from this matching, so it favors mismatch. And so here just an example, so how trait values are distributed, this for the host and this for the parasites. Here is, for example, the environmental optima, and this would be environmental selection because they want to match the optimum trait. And in the case of selection, the parasites, they want to match the traits of their hosts, and the hosts, they want to mismatch. Basically, this is how we implement these kind of models. And we parameterized it in different scenarios. These are synthetic landscapes. So we are changing in different degrees of forest cover, going from 10 to 70%, and different degrees of the level of the fragmentation in the landscape. And we also classified each one of those patches if it's a core habitat, so it's completely surrounded by forest patches, if it's an edge patch, or if it's a disturbed patch. So with this, we had 12 different scenarios changing the configuration and the fragmentation of the landscape. And we use this data is the regional meta-network of hosts and parasites, so mammals and bacteria and viruses, parasites. This is summing 51 hosts and 103 infectious agents. This is how the meta-network looks like. And for parameterizing the frequency of occurrence in forest, that is the parameter of the host to indicate if it's favored by forest or not, we use the number of occurrences in forest area divided by the total number of occurrences that we had of the different species. So here are some results. This is the different scenarios changing the forest cover. This in the different type of patches, if it's core, edge, or disturbed patches. And here is a measure of the local network structures that we have in each one of those local patches. And this first component of this PCI of the structure is indicating that if you have higher values, then you have smaller and more connected networks. And what we found is that local networks in disturbed habitats, they are much smaller and more connected than the local networks that we find in core patches. And also that increasing forest cover, we tend to also increase the size of those networks and make it less connected. When we look at the within habitat dissimilarity among those networks, so this is a measure of the beta diversity of the networks, let's say. So this is a measure of the dissimilarity among those networks. We see that for the core patches, they tend to be pretty similar, and it's not really affected by the degree of forest cover. But the disturbed and the edge networks, they tend to be more dissimilar among them. So you have a higher heterogeneity of interactions there. And the ones in disturbed patches are the ones that are more affected by forest cover. When we look at the consequences for the ecological dynamics, here in this plot, I'm showing a measure of relative degree of the parasites. What this measure is the local degree, is the local host range divided by the regional host range. So the higher is this value, the more similar is the host range local and regional. And the lower mean that local, so the local networks, the parasites interacting with the smaller subsets of all the potential hosts that it can interact with. So, and this is the relative host abundance in total. So we can see also that the type of habitats greatly affect this, because we have a much lower relative degree in disturbed habitats, because they are interacting with a subset of all their potential host species. And when we go to more conserved core habitats, they are interacting with the entire potential host range that they have. And also, well, if this increases relative abundance also. And this was particularly true for the zoonotic viruses and bacteria. So they highly increased this measure of a relative degree in disturbed habitats. And finally, the consequences for the evolutive dynamics. Here I'm showing this measure of trait matching. The trait matching would be the degree of matching of the parasite with the host species. it is a measure also of local adaptation. Let's see if they are more adapted or less adapted to the hosts. And this is the network structure. So there is there is an effect of network structure on the trait matching, and there is a higher trait matching between parasite and host species in the disturbed patches than in core habitats. So smaller and connected networks are enhancing the matching among parasite and host species, and the abundance of host species also impose important selective pressure there. So to synthesize the results, we found that the type of habitat is shaping the structure of the local networks. So disturbed patches, they have smaller, more connected, and very heterogeneous networks. The edge habitats, they are more nested with a composition of hosts and parasites that is more similar to the core environments. In the case of the core environments, they are more homogeneous across the landscape. They are larger, and they are more modular local networks. And considering the evolutionary dynamics, so the realized host range of the zoonotic parasites is varying more across the local networks in the disturbed patches, and trait diversity increases the adaptive potential of the parasite species in fragmented landscapes, because we found these effects in the trait matching. And it's interesting because those previous results using models to try to reach measures of parasite probability of occurrence and dynamics and prevalence, they were very much theoretical models. But when we investigated the effect of different parameters in the prevalence of these zoonotic disease, that is the bubonic plague epidemics in Brazil, is very interesting, because we found that parameters related to the host parasite networks, so the connectance and the degree and the interaction dissimilarities, were the factors that had a stronger effect in the number of human plague cases, and even stronger effect than other parameters that are more closely related to the cycle, epidemiological cycle, and episodic dynamics of the disease. So what this is indicating us is that the networks, they can be a very important predictor of the susceptibility of the different places to the emergence of different diseases. So with these results, we were trying then to investigate different aspects of land use change affecting the parasite-host interactions. So considering capacity and opportunity space, we found that there is enhanced parasite adaptability and diversification, which could increase probability of sharing. Fragmentation is favoring key reservoirs, is changing the network structure, and is also increasing the variance of those realized host ranges. And those are very interesting and potentially useful measures to infer potential probability of emergence in these. But it's also true that if we want to better understand what is the risk of disease emergence, it is also very important to consider aspects that are not only related to the hazard, that is the diversity and prevalences of parasites and the contact and transmission, but it's also important to consider factors that are related to different vulnerability of the human individuals and populations. Because even if you have a higher load and hazard, let's say a higher probability that you're going to have high prevalence of parasites, the probability that a person is going to get infected is going to depend not only on that, but also on historical, economical, and political factors that is related to their immunocompetency and their exposure and several factors. So as a first approach for trying to also incorporate those different risk components that are related to exposure, vulnerability, and coping capacity. We investigated all the zoonotic diseases that we have in Brazil. This is the distribution of it across the territory. And we investigated the effect of all those different variables of risk category. And those are the variables that were significantly related to the mean cases of zoonotic diseases distribution. And actually, yes, we did found that natural vegetation cover was the stronger predictor of zoonotic diseases in the, across the territory. Also, the richness of the different mammal species, but it's also true that different components related to the social component of the landscape, such as the remoteness of the city, so this is related to mobility, and also the structure of the cities, how is the urban vegetation and vegetation loss. Those were also parameters, variables that were related to the number of cases. So the research I'm now doing is trying to explicitly incorporate those socio-ecological parameters in the network framework in order to better understand not only the effects of those drivers, but also the potential feedbacks among social and ecological processes. And then for doing this, the general framework we are trying to develop is linking land cover in use with the different productive systems of the landscape. So this is depending on the different ways that the economy is organized in each one of the places. We have different productive landscape units, and this is in somehow also related to the mobility and trades, to the interactions among humans and nature, and with the local host parasite network. And with this framework, we expect to better understand the feedbacks between the social and the ecological processes related to vulnerabilities. And with that, I would like to thank you very much for the audience and thank all so the postdocs and PhD students in the group and the funding agencies. And if I can take some questions, I would be happy. Thank you very much.
Ahora, sí. If anyone has questions, please go ahead.
First of all, thanks for the nice talk. And of course, this is very complex, so I can understand. So my question will be a little bit naive in that sense. But basically, you are mixing within the umbrella of parasites a lot of things that are very different, with very different life cycles. And in particularly, my question is about whether your models will improve if you consider the dynamics of all these invertebrate vectors that are in between your vertebrate hosts and your parasites, like fleas, ticks, or mosquitoes are not the same, so they have very different dynamics. So that should influence perfectly all the movement of the parasites, their diversity. Yeah. So we are indeed trying also to incorporate this. It increases the complexity of the models. But in the bubonic plague example, for example, we are considering the flea species and how they are connecting those different host species. And for doing that, then we have to, as a first approach, focus in one disease system. And we also did that with Chagas disease because they have different mechanisms of transmission. You have trophic transmission, and you also have the vectorial transmission. And so I think it's focusing in one system, and then you have more accurate predictions for that system, but as a broad overview, less
general, because also those different diseases, as I showed previously, they have land use changes as a main driver. So we wanted to understand in the macro ecological perspective. But I agree with you that if we want very accurate and local predictions, then we have to focus in a specific disease system.
Okay. Thank you for the talk, Cecilia. That was nice. I would like to go back to the first networks you have shown in your presentation and ask you a couple of questions. You said you were, okay, that was so many slides. You measured centrality.
This one?
In those networks, yeah.
Yes.
And you were correlating your measure of centrality with some other quantities. Which measure of centrality?
Betweenness centrality.
Betweenness, okay.
Yeah.
Have you, are all the networks here, do they have the same size?
So this is one, this meta-network, so this was not local networks, they were different approaches. So in this case, we had local host communities. So the hosts, they changed their occurrence in the landscape, so they have different probability of occurring. Right. But this network were only to classify if the hosts, they were central and non-central, but according to the general meta-network of the biome. So
okay, so it's just one single network. That's nice, yeah. Because otherwise, I mean, as you know, between the centrality would change depending on the size, right?
Yeah.
Okay. And another question has to do with your models, whether you have fragmentation, habitat fragmentation.
Yes.
And they're really nice, and they're considering many aspects of this fragmentation. But as you know, also there is a transition to percolation in your available habitat.
Yeah.
Or when you decrease or change the parameter of your fragmentation. I have not seen this signature in your results. They seem to be pretty linear without large dispersions, but pretty linear. So could you comment on that?
Okay, let's go back to those results. So you talk about this one, no? That we don't see this here.
Yeah, in general.
Yeah.
I haven't seen it. I mean, I was looking for it. So I haven't seen any signature of this transition. So you should be transitioning from a fragmented network with average increasing size in the isolated components to a network which is essentially a main connected component with isolated.
Yeah, this we did found in the. It's because here they are. Well, you have the variance there, but I'm just showing the mean. But well, here more or less you have this different separation, and in the case of this evolutionary one here, then you do have this, but well, there is this.
Right.
It's not linear.
Yeah.
It's not linear, but there is this shape.
Yeah. So I mean, I would expect here something essentially flat, and then a rise, a significant rise after percolation.
Well, maybe here.
Somewhere, I don't know.
You know that this percolation, now I am with another, postdoc in the group, and he's actually trying to understand percolation phenomena in these.
Right.
And then we see percolation, we do see.
Yeah, then also, I mean, the size of the system plays a role.
The size of the system is a crucial role. Yeah,
okay.
This size is fixed.
So that's good that you take into account these variables also.
Yeah.
Sure. Okay, thank you very much.
Thank you.
Thank you. Just to follow up on this question, in the x-axis, in the fragmentation, is this the percentage of fragmentation?
Percentage.
Because as far as I know, usually the percolation transition happens at 0.6, like 60%, and maybe you didn't reach, or maybe you're just starting, or not.
Finite size effect.
Sorry? Finite size effect. Okay.
Yeah.
Okay.
Yeah, have to increase the
yeah. We have another question from the streaming. Someone is asking, I wanted to ask what data source you use for mammal richness and what data source you use for land use. Also, another comment, cool presentation, keep it up.
Okay, so I think it's for For the meta-network or for, well, no, going back. There is this paper here, okay. So the occurrences is a database that is published in this paper that are the local here. The local communities in each one of these places, the local communities of the different host species. This is in this paper. And the network is coming from this other paper that we have. It's a data paper, and it's this one. It's the BMPO, and this is for the entire Brazil, but it's separated also by biomes. And this is Cruz et al. Is a data paper. So this is where the data is coming from. And the occurrences, the broad one, this is coming from this database that is from the Ministry of Environmental Ministry. So they have all these occurrences. And the land use is map biomas. This is available online also.
Any other question? No. I have a curiosity. You showed that co-occurring species, they are still not sharing potential parasites. Do you know if it's related to phylogeny limitations or something? And also, I have no idea about this topic, so is there any time expectation about how much time the parasites need to parasite a new host or something like that?
There is one factor that I think maybe it's the most important one, is related to limited sampling, because maybe it was not yet sampled, this association, because we need more data, and this was also highlighted in this paper. We don't have this association registered, but because the species is rare or is very low abundant, and then we don't have many records of parasite associations to this species, so this is one limitation. And other limitation, I think, well, phylogeny. Yeah, but this group is pretty much, we were just looking at the rodents and marsupials, so it was I mean, phylogeny there, I think it's not exactly restricting, and I think it's can be related to abundance. If they are rare species, so you don't have, you didn't give it not yet the opportunity that they co-occur, but maybe they are not exactly interacting, and I think it's related to that. But if you change the circumstances of the place, and then maybe you favor one species, it increases in abundance, then you start having potential contacts, and you can maybe have a transmission.
Okay.
Hi, thanks. It was really, really fascinating talk. So you showed, I think, that the fragmentation was having a bigger effect on the host ranges for the viruses relative to the bacteria. Is that correct? In the proportion of the host range they were covering. So you showed you're going to have to go between your slides again. You showed a graph that showed the RNA viruses above, and then okay. So is that essentially because the viruses are doing everything more quickly, or responding to the available hosts more quickly, changing their host use more quickly in the disturbed patches? Or are there some other traits which are influencing the changes in host use? And does that make the viruses more dangerous as zoonoses in terms of when human population is exposed to them than the bacteria, for example?
Yeah, in this model, we don't have any differentiation, let's say, among those species. So all of them have a trait, and there's nothing that is telling the model, this is a virus or this is a bacteria. The only thing that they differ is in the connections, so it's in the network of connections with the potential hosts that they have.
So the viruses tend to have more potential connections.
Okay. Yes, and they also lose. So the host species that they are losing locally, maybe they are losing more hosts locally, and that's why they are getting more heterogeneous across.
And does the local differentiation make them more susceptible to be zoonoses, essentially, or do they make them simply more difficult for health infrastructure to deal with, or something like that?
There is one thing that I think is difficult to answer. That is, well, we have the patterns of associations, and it's clear that the zoonotic pathogens, they are interacting, associated with those host species that are more generalist, and they are highly favored by changes in the landscape, in the environments, and all these. But I am always asking myself, is this a consequence of is it like this because they are zoonotic, or are they zoonotic because they are specifically favored? And I think that maybe it's the second one. I mean, they become zoonotic because they are being favored by these ecological and evolutionary processes, and then they become zoonotic. So, but I don't have a concrete answer for that.
So that's related to your map showing that there are more zoonoses detected in Brazil than anywhere else. It's partly because there's a high diversity of hosts and a high diversity of microorganisms, but also because of the fragmentation, it's increasing the risk that they'll become zoonotic, and also that we'll detect them, I guess.
Yeah, but again, also there is a great sampling bias and data limitation. So in general, those macro patterns, we have also to be cautious when considering these. But yes, it's mostly related to the diversity of potential hosts.
Great, thank you very much.
Thank you.
Any other question? No? Well, thank you a lot, Cecilia. It was great. And thank you all for coming.
Thank you.